Good day, everybody. So my name is Tuomo Mantere. I'm a postdoc. I worked at the Immunogenomics and Genomic Technologies Research Group at the Radboud University Medical Center in Nijmegen in the Netherlands. And currently I'm working at the University of Oulu in Finland in the field of cancer genetics. So today my talk will be about the cytogenetic assessment of 52 leukemia genomes by bionanogenome imaging. So we obtained the bionano Saphyr system in Nijmegen in early 2019 and had the training in March 2019. And since then we have ongoing research projects, mainly focusing on detection of somatic structural aberrations in leukemia and compared to the standard of care diagnostic test results, including FISH, karyotyping, and CNV microarrays. And then we have another ongoing study in collaboration with our dear French colleagues, and there will be, this is about the detection of structural germline cytogenetic aberrations, and there will be another presentation by Dr. Laila El-Khattabi on this. And finally, our third approach has been to use bionanogenome imaging to solve some of these really difficult to solve rare disease cases. And there is another presentation by Dr. Alexander Hoischen on this topic in the same symposium. So, as many of you know, the comprehensive analysis of all structural variants in a given leukemia sample, it still requires a combination of different techniques, including traditional karyotyping, FISH analysis, and CNV microarrays. So in a diagnostic setup, it would be highly beneficial to have a generic single cytogenetic test to use instead of the combination of all these three. So that in mind, we set up proof of concept study where we included 52 leukemia samples that were analyzed with the standard of care diagnostic cytogenetic tests, including microarrays, karyotyping, and FISH. And these 52 leukemia samples, they consisted of a mixed bag of different types of leukemias, most commonly MDS and AML. And the leftover material after the standard of care tests was used for genome imaging with the Saphyr system. And then based on the diagnostic test results, we divided our cohort into simple cases which had less than 5 diagnostically reported aberrations, and to complex cases which harbored 5 or more aberrations or unspecified marker chromosome. And the main source of material in our cohort was bone marrow aspirates, about 80% of samples, and then rest were from peripheral blood samples. And we always isolated the ultra-long DNA molecules from 1.5 million cells and performed the labeling using the latest DLS chemistry. And the samples were then run with the maximum capacity of 1300 gigabase pairs of the current chips that we are using. And then a little bit about the technical summary for our study cohort. So indeed, we had about 1300 gigabase pairs of data per sample, and the molecule length, the length of the molecules that were used for the analysis on average was 260 kilobases, and the map rate about 72%, resulting in effective coverage of 280-fold per sample on average. So really high coverage. And as many of you here might know, that there are 2 different analysis modes in access to analyze bionano data. So the de novo assembly-based variant annotation and SV and CNV calling. So this is what we use for germline constitutional variants. But in this study, in this leukemia study, we used a rare variant pipeline for SV and CNV calling, which is specifically meant for detection of somatic structural variants. And so how much do we find SVs and CNVs per sample is a question we often encounter. So on average, there is 13 high confidence SV calls that are larger than 100 kb per sample. And this is after excluding all the common germline SVs using a population control database. And 10 of these SV calls are overlapping with a gene. And quite many of these calls are from result from large rearrangements such as interchromosomal translocations or large inversions or interstitial deletions. And for CNVs, there is a separate algorithm calling CNVs. So on average, there is 8 gains and 47 losses per CNVs per sample, called as loss. And some of these large, this number may seem rather high, but it's good to note that some of these large CNVs or aneuploidies are over segmented. So actually there's like one single big CNV is called as multiple different ones. So these numbers are somewhat inflated. But of course, we also have really highly complex and rearranged samples that do harbor a lot of CNVs. And then the most important question is that how are we concordant with the standard of care results? So are we able to find back all the aberrations that were previously picked up by the diagnostic tests with the classical approaches? So I will start with the simple cases. We had 36 simple cases, as I said, and these harbored in total of 46 aberrations that fall within the scope of this study. So for now, we had to exclude the copy-neutral loss of heterozygosity and structural variants, that balanced structural variants with centromeric breakpoints and structural variants with the variant allele fraction less than 10%. But in total of these 46 aberrations, they were most commonly we had deletions and different types of translocations, but also aneuploidies, inversions, and duplications. And important result here is that we find them all back. So we reached 100% concordance for the simpler cases. We didn't miss any of the previously detected aberrations with using bionanogenome imaging. And for the complex cases, we've reached the full concordance for 15 out of 16 of these cases. And one of these cases that which actually had multiple different aberrations, there were three CNVs that we were not able to pick up with genome imaging, but all of these aberrations actually had variant allele fraction around 10%, which is just the borderline of what we are able to detect with the current setup. And generally for the complex cases, it seems that the optical mapping is able to more fully resolve the true underlying genome architecture compared to the previous tests alone. So I will show some example later on this. And in addition, we are able to find a lot of novel new aberrations that were not picked up with the previous tests. For example, balanced translocations. And this opens up a lot of possibilities for new research as well. So then I will move on to some examples. So one important class here are the simple, in this context, small deletions that are hitting a well-known leukemia genes. So for example, previously in this sample, there was a 700 kb deletion hitting the TET2 gene picked up with the CNV microarray. And we run this same sample using a bionano. So in this circus plot view, you can already see that there is, this is chromosome 4. So we have a CNV and an SV call hitting the TET2 locus. And when we zoom into this, we can clearly see that this deletion is called by both the coverage-based CNV tool and also with the assembly-based SV tool. So both of the algorithms are able to pick this up nicely. And then moving on to one of the really strong points of BioNano, and especially important class of variation in leukemia. These are the balanced translocations. So in this complex sample, using a combination of karyotyping and FISH, they detected a translocation between chromosome 11 and 14, which results in this well-known IgH CCND1 rearrangement, which is commonly seen in leukemia. So we run this with the BioNano, and in this circus plot view of chromosome 11 and 14, we immediately picked up this translocation between these two chromosomes. And when we zoom into this event more closely, we can right away see that the genes IgH and CCND1 are involved in this rearrangement. So this is nicely concordant with the previous tests. And I mentioned earlier that we think that we can find a more complete picture on many of these complex samples when we run it with BioNano. So for example, this same sample I showed before had a multiple CNV calls with the CNV microarrays here. For example, these gains and losses of chromosome 3 and chromosome 8. And when we run this with the BioNano, we can see that actually in quite many of these complex samples, we see that the findings from the previous CNV microarrays are mediated by previously undetected translocations, as here. We can see that the gains and losses in chromosome 3 and 8 these breakpoints are matching exactly with the breakpoints of these translocations that were previously undetected, despite the use of karyotyping. So moving on to a chromotripsis case, we had this sample with the karyotyping, with the combination of karyotyping and CNV microarrays, we're able to pick up that there is a chromotripsis of chromosome 8. And once again, with the BioNano here, with the circus plot view, we can see that the circus plot is quite clean for other chromosomes. But for chromosome 8, it's just full of large rearrangements and complex deletions and inversions and everything. And the CNV profile is also, if you look at this figure, it's matching quite nicely with the CNV microarray on top and the BioNano CNV profile on bottom. And it's matching really well with what we see with the CNV microarray. And then this is a well-known recurrent translocation between chromosome 9 and 22, resulting in the BCR-ABL fusion, which is a commonly seen aberration in CML. So in our cohort, we had 3 CML cases with this BCR-ABL fusion. And with BioNano, we were able to pick up all these 3 events. And on the left, we can see that there are 3 different predicted breakpoints in ABL gene and 2 different breakpoints in BCR. So it seems that the breakpoint resolution is much better than in traditional FISH also. And then one additional thing we did for our samples was we tried to look for balanced translocations that could lead to potential novel fusions. So what we did, we checked for all the balanced translocations and that are involving a known cancer gene from Cosmic database, and which were not reported in our dataset with the previous techniques that were used. So we identified some of these as well. One of them was this IgH-BCL3 translocation involving these 2 genes. This is actually known in literature, but it was previously undetected in this sample. So, and it could be really clinically important translocation as well. And then there is one other MSI2 UBE3C translocation involving these two genes. And this is actually really interesting for research, since this was the only aberration that was detected in this sample, and it might result in a fusion, novel fusion that could be a potentially a novel driver of leukemia in leukemia. But of course, this requires a lot of follow-up still. But overall, I would say that Bionano can be used as an efficient tool for mapping novel putative fusions. And then also about the copy-neutral loss of heterozygosity detection. So currently, this is not yet enabled in the tools that Bionano offers, but we manually take a look at this sample where we knew already that there is a large region with the copy-neutral loss of heterozygosity in chromosome 7, and we just manually checked the zygosity of all the SV calls in these regions and compared these with the region that is not supposed to have a loss of heterozygosity. And we can quite easily see from here that the heterozygosity is lost in this sample, in the matching the CNV array results very nicely. And then I would like to conclude, just summarizing a little bit. So we had 52 leukemia samples, and these had previously been tested with the standard of care cytogenetic tests, including karyotyping, FISH, and/or CNV microarrays. In total, this consisted of 36 simple cases with less than 5 reported aberrations, and then 16 complex cases with 5 or more aberrations. And for the simple cases, we were able to find all the previously reported aberrations with a really low false positive rate. And for the complex cases, for 15 out of 16, we found all the aberrations back. For one case, we missed some aberrations with low variant alpha fractions. But overall, the complex cases seem to be even more complex than previously thought based on the previous tests. So the genome imaging, it's likely closer to the true underlying genetic architecture in these samples. And the next step for us is to perform a prospective study for leukemia in early 2021, where optical mapping and standard of care tests are run in parallel and compared. And then I would like to acknowledge especially Cornelia Nevling and Alexander Hoyschen, who have been really the driving force behind this study, and many other colleagues, also Marianne, especially for help in the classical cytogenetic approaches, and many other colleagues and the people from Bionano for their help in many aspects of this study as well. Thank you.